Today we are going to study food testing, biochemical testing of carbohydrate, different types of carbohydrate and proteins and fats. So first of all, we will see the starch. We will see the chemical test and its observation. So what is starch? Starch is a polymer of glucose. Usually plants convert glucose into starch, that is the storage form of carbohydrate in plants. It is insoluble in water, or you can say that it is partially soluble in water. It makes suspension in water. What to test or how to test is we will take a test tube and a little amount of warm water or the cold water can be taken there. Then on shaking, a suspension of starch is produced. Then we will add 3 to 4 drops of iodine solution. So the iodine solution that is brown in color, it turns blue black that you can see here. See, this is the brown solution of iodine, and see what happens when we add the solution into the starch suspension. So this a few drops of iodine solutions are added here, and on shaking, a light gray color appears. But I think the quantity of iodine is not too much, not sufficient. So when we add a few more drops, then the color becomes clearly blue-black. So it shows the presence of starch. So wherever you are uh, going to detect a sample that whether carbohydrate is present there or not, you can use this method. You just use a few drops of iodine, so it will gives you blue black color. The color intensity depends on the quantity of starch as well as the concentration of iodine solution. Next is the reducing sugars. Some of the sugars, for example glucose, fructose, maltose, these are the examples of reducing sugar. They have capability to reduce certain uh, certain other reagents. For example, in this case, copper is reduced. So, how to perform this test? Uh, we should take about 2 centimeter cube or the 2 ml glucose solution in a in a test tube. Then we will add an equal volume of Benedict solution or the Fehling solution A and Fehling solution B. We can use either of these reagents. And then we place the test tube in a beaker of boiling water bath to heat it up to 4 to 5 minutes and sometime little more. This test can be carried out by direct heating instead of using the water bath. So what happens? See, the solution changes from clear blue to cloudy green, then yellow, and finally to red precipitate. And these red precipitates are actually copper 1 oxide. Uh, Actually, the reagent that we are using here, Benedict solution or the Fehling solution, the main the main ingredient of the Benedict solution or the Fehling solution is copper sulfate, where copper 2 ions are there. So copper 2 ions, where copper is in 2 plus charge, so copper 2 ions are converted into copper 1 ion. That is the reduction of copper 2 ions into copper 1, and they form the oxide. So as a result, we obtain either the cloudy green, yellow, or red precipitate. So It shows the presence of reducing sugar. Remember that reducing sugars are not only the monosaccharides but also the disaccharide, for example, maltose, lactose. But sucrose is a non-reducing sugar. If you carry out this reaction with sucrose, then the blue color of the Benedict solution or the Fehling solution remains there instead of giving the precipitate. Now see the video. This is the real demonstration. Here we perform this test by using the Fehling solution. This is Fehling solution A, that is 1% copper sulfate. And see, on adding this copper sulfate, nothing happens. But when another solution, that is Fehling solution B, that is the mixture of sodium hydroxide and sodium potassium tartrate, then it becomes dark blue in color. And after heating, you can observe the green, yellow, or red precipitate. Now see, that copper sulfate is added, that is Fehling A solution. Now the Fehling B solution, that is sodium potassium tartrate and sodium hydroxide. Now it becomes dark blue, and now we will put it in boiling water bath. And during this, you will observe that the color changes. Now see, a slight change is happening here. It is becoming green, and now it will be changes to yellow in color. And finally, we will get the red precipitate. And we can use the electric water bath as well. It depends on the laboratory condition, whether you have well-equipped laboratory or the simple school laboratory. So now you can see that red precipitates are there, and on the top there are the green precipitates. If you will continue to heat, then all would become red. Now see the same test with Benedict's reagent, and here we are using the direct heat, but not necessarily that we use the direct heating for Benedict's reagent. We can use the direct heating when you are using Fehling solution as well. So it is not specific. Now you can see this is the Benedict solution, and we are adding the Benedict solution in reducing sugar, solution of the reducing sugar. It is glucose in this case. And see, on heating, there would be color change. Now you can see that a slightly green color appear. And now for further heating, it is put on the flame. It is becoming yellow. See, it has become yellow. And finally, it would be converted into red color, red precipitate of copper monoxide. So you can see this. And now see the test for proteins. All types of proteins can be tested. That is name as the name of the test is Biuret test. So we can take the sample of protein as, for example, flour of the pulses or meat extract or egg white, solution of the egg white that actually makes a suspension. So how to uh, how to perform this test? You just take the 2 centimeter solution or the suspension of protein in a test tube. Then add a few drops of 0.5 mol per decimeter cube solution of sodium hydroxide and shake it well. Then add 1 centimeter cube, 1 ml solution of 1% copper sulfate solution that is blue in color. Then put the test tube in test tube stand for 1 to 2 minutes, or you can hold in your hand. So the blue color of the copper sulfate turns into purple or violet color. This test is known as biuret test. Now you can see the video. We have taken this protein suspension in the test tube, and then we are adding sodium hydroxide. And after adding the sodium hydroxide, we are adding a few drops of copper sulfate. Copper to sulfate is added here. So, so the color remains same, but after a few minutes, it becomes purple. So whenever protein is present in a sample, it will give the purple color. And again, the color intensity depends on the quantity of protein. Now the test for fats and oils. It can be tested in two ways. First, you can take the sample, either the butter, cooking oil, or egg yolk, which have fats. Then the test, take a test tube and add a few centimeter cube of water. Take another test tube and add about 2 to 3 centimeter cube ethanol or acetone. These are the organic solvents. Then add 2 to 3 drops of cooking oil or any sample that you have, and shake well to dissolve this fat sample into the acetone or ethanol. Then pour the solution into the test tube containing water that we have first taken. You can see the first step, and on shaking well, we get the milky white emulsion. Now see, in this video, first we are taking acetone. This is acetone, and in the acetone, this is acetone, about 1 to 2 ml, and then we have added a few drops of cooking oil, and it is shake well. It dissolves, and on adding water and shaking, it turns milky. You can see now it becomes milky now. Now you can see another another way to test fats. Here we are using ethanol and cooking oil. So what is the change here? You can see, you can observe here that first we have taken a small quantity of oil. Even you can take this later on adding ethanol, and when we shake it, it becomes milky white. Actually, why it happens? Because water is already in this commercial grade ethanol. So that's why this emulsion, this milky white emulsion formed directly, because water was already in this commercial grade ethanol. So this is also a test for fats and oils. So you can use either of the way to check the sample whether it contains fat or not. Now see what are the applications of these food testing. We can use these tests that we have seen in this video for sample of food like bread, milk, potato, cassava, resins, onion, beans, or any other food sample to test whether such materials reducing sugar, starch, or proteins, or fats or oils are present there or not. So, how to perform the test with these samples? So, first we will make the sample. In case of proteins, we crush the solid sample, proteins or carbohydrate, any type of carbohydrate. We crush the solid samples in a mortar and shake with warm water to get a solution or its suspension. Then we will pour the small amount of the watery mixture into several test tubes because we have to perform tests for all types of food material. But to test a lipid, the food must first be crushed in ethanol because lipids are soluble in water, not soluble in water, but soluble in ethanol or acetone. So here you can use acetone as well. And then we will filter it so that only the fat sample will be transferred. And then we perform the routine test that we have seen in our video. Test the sample for starch, glucose, or proteins as per protocol discussed. And for lipid, for the clear filtrate that we have obtained here in this last step, you can see, and then you will check. So if you will get the milky white emulsion, then the sample contain fats or oil. Or for starch, glucose, and protein, you have seen the same result would be appear if these are present in your food sample.